Okay, so in this second video, we are going to discuss on part B of chapter 3, which is on plasma membrane transport. We are going to look at 4 structured questions. So for question number 1A, figure 2 shows a type of active transport in plant. Okay, so you have here two transport protein. The first one is proton pump. We call this one is as proton pump. The function of this proton pump is to pump hydrogen ion from inside of the cell to the outside of the cell. So as you can see, inside the cell, the concentration of hydrogen ion is low. So to move hydrogen ion from inside to outside, from low concentration to high concentration, it requires energy from ATP molecule. Okay, so the hydrogen ion is pumped out through the proton pump and causes outside to have a high concentration of hydrogen ion. So now, the purpose of this is to concentrate hydrogen ion in this region. Okay, in this region, so that the sucrose outside of the cell can enter into the cell along together with the hydrogen ion through this transport protein, the second transport protein or carrier protein. We call it as hydrogen ion sucrose co-transporter. Okay, so it leads to question number 1 here. Name carrier protein S, okay, which is hydrogen ion sucrose co-transporter. Okay, so as you can see, outside of the cell, the concentration of sucrose is low. So sucrose needs to enter into the cell through this co-transporter from low concentration to high concentration inside cell. So basically, this type of transport protein occurs in plant. Okay, so sucrose needs to move into the plant cell so that the plant cell can accumulate, okay, a high concentration of sucrose in the cell. Okay, so it means that for example, this one, it can involve the cell of vascular tissue. Okay, so vascular tissue. So sucrose will be transported from the leaf of the plant that produces the sugar molecule, and then the sugar molecule from the leaf will be transported via the vascular tissue. So it needs to be transported from low concentration to high concentration. So the plant cell needs to have a high sucrose concentration inside cell. Okay, so the movement is a type of active transport. Okay, active transport because it is from low concentration of sucrose to high. So in order to bring sucrose into cell, okay, it requires hydrogen ion to move along together with it, okay, through this co-transporter. So remember, okay, so when writing the name of this co-transporter, you always have to write the ion first. Okay, the ion first. So in this case, the ion is hydrogen ion. So hydrogen ion sucrose co-transporter. Co-transporter is referring to this transport protein. If you just write hydrogen ion co-transport is the process. Okay, is the name of the process. Okay, by adding co-transporter ER at the end, so it is referring to this transport protein. Okay, co-transporter. So hydrogen ion sucrose co-transporter, remember to put the hydrogen ion first, okay, and then the nutrients, okay, to be taken into the cell, which is sucrose. Okay, so B, describe how sucrose is transported in plant. Okay, so in this video, okay, in this slide, I'm just writing or giving you the key point. Okay, so you in the exam, for example, you have to write the whole sentence explaining your answer clearly. Okay, not in point form like this one. Okay, so okay, so describe how sucrose is transported in plants. So the mark is 3 marks. Okay, so for the first point, you have to mention the role of the proton pump here. Okay, so which is the function of the proton pump is to pump hydrogen ion, okay, across the membrane from each side of the cell okay, to the outside of the cell. Okay, from inside to the outside of the cell. Okay, so that you get 1 mark. And then after that, what happens? Okay, so this will cause hydrogen ion concentration outside to be high. Okay, high hydrogen ion concentration outside, or a high electrochemical gradient. So this will cause, okay, electrochemical gradient to be generated. Okay, electrochemical, only one M here. Okay, so electrochemical gradient, high electrochemical gradient will be produced. Okay, so therefore this will generate a form of energy. We call it as potential energy. Okay, potential energy will be generated, and this energy, okay, will be used by the co-transporter to transport sucrose from outside into the cell. Okay, from outside into the cell. Okay, so next, okay, so this one, any of this point, you will get 1 mark. Okay, so you can explain up to this point, or you can elaborate more for your understanding. Okay, so the hydrogen ion sucrose co-transporter causes the diffusion or the movement of hydrogen ion, okay, into the cell, which is down electrochemical gradient. So the movement, okay, from high to low electrochemical gradient is a type of potential energy, okay, that the co-transporter used, okay, to bring in the sucrose from outside to inside, from low sucrose concentration to high concentration gradient, or against concentration gradient. Okay, so this will cause the cell to uptake sucrose into the cell. Okay, so each of these points will worth 1 mark. Okay, 1 mark. Okay, B. Figure 3 shows the mechanism of cholesterol transport across cell membrane. Okay, so name the mechanism. So mechanism of transporting cholesterol from outside into cell. So this is the outside region. Okay, outside region. So this is inside cell, the cytoplasmic region, and this is the plasma membrane. Okay, so along the plasma membrane, you have receptor protein that will bind to cholesterol outside of the cell. Okay, cholesterol outside of the cell needs to be taken into the cell by cholesterol, or the ligand we call it, to bind to the receptor protein. Okay, so the region that has the receptor protein here, as you can see, inside it you have a layer of protein inside. Okay, inside the plasma membrane. Okay, lining the membrane. Okay, lining the membrane. So you have a layer of protein. Okay, a layer of protein. So inside you have a layer protein. Outside you have the receptor protein that binds to, with the cholesterol, or the ligand. Okay, so once the cholesterol binds, okay, to the receptor protein, it causes the membrane to fold inward, and this we call it as formation of coated pit. Okay, so coated pit will then form coated vesicle that brings in cholesterol, okay, in bulk into the cell. Okay, so this is a type of bulk transport actually. Okay, so altogether we call it as receptor-mediated endocytosis, which brings in specific molecule into the cell in large quantities. Okay, in bulk. Okay, so describe question 2. Describe the above mechanism, which is you need to describe the receptor-mediated endocytosis, worth 3 marks. Okay, so okay, so for the first point, okay, so cholesterol, or the ligand is the chemical molecule that bind to the receptor protein. Okay, so in this case, in this question, for instance, so it is the cholesterol, or the ligand, bind to receptor proteins that are found on the plasma membrane. Okay, so facing the extracellular fluid side lah, that is. Okay, so so the binding of cholesterol to the receptor protein will cause the plasma membrane to fold inward. Okay, to fold inward. So it starts to fold inward. Okay, forming coated pit. Okay, it starts to form coated pit, and from coated pit it will form coated vesicle. Okay, so the formation of coated vesicle containing cholesterol will cause cholesterol to enter into the cell. Okay, cholesterol are brought into the cell, and then after ingestion, okay, after the cholesterol is being ingested into the cell, okay, so the cholesterol will be released from the cell from the vesicle for the cell to use, okay, for metabolism. And then after the cholesterol is released from the vesicle, the receptor protein, okay, the receptor protein here can be recycled again. Okay, so the receptor proteins are recycled back to the plasma membrane. So any of these three of this point, okay, any of these three will gain you 3 marks. Okay, so next question, differentiate, okay, the mechanism in B, which is receptor-mediated endocytosis, with exocytosis. So endocytosis, it brings in, okay, specific molecule into the cell. Okay, brings in. As for the exocytosis, it the process will bring out or secrete out product from cell or waste from cell to the outside of the cell. Okay, so the difference, for the first difference, is that for receptor-mediated endocytosis, cell takes in or import biological molecule or nutrients. So in this case, is the cholesterol, for example. Okay, so as for the exocytosis, cell takes out or secrete out or export out, okay, biological molecule or cell product, okay, from inside to the outside of the cell. Okay, so the product or wastes from the cell even. Okay, so that is the first difference. Okay, and then for the second difference, for receptor-mediated endocytosis, so it brings in specific molecule into cell. Okay, because due to the presence of receptor protein. Okay, so it brings in specific molecule into the cell due to the presence of receptor protein. And as for the exocytosis, the process is non-specific. Okay, non-specific, or for exocytosis is it does not require any receptor. Okay, no receptor involved. Okay, so the total mark for question number 1 is 10 mark. Okay, so next question 2, give one function of membrane protein. Okay, so for this one, if you refer back to your notes, the role or the function of membrane protein, there are 6 different function of protein. So the membrane protein that are embedded in the plasma membrane can become enzyme. Okay, enzyme, the first one. Okay, you only need to list out one. Okay, so it can become enzyme, or it can become transport protein, or it can involve in cell-cell recognition, or it can involve in signal transduction, or it can involve in okay, as you can see here, okay, in intercellular joining, or in the attachment, okay, to the cytoskeleton and extracellular matrix. So any of these point, okay, or role of the transport protein is correct. Okay, so you need only one, okay, one to be listed in your answer. Okay, transport protein, enzyme, signal transduction, cell recognition, intercellular joining, such as the desmosome, for example, and then attached to the cytoskeleton, and also ECM, okay, to enhance the structure of the membrane. So this one is only applies for animal cell, as animal cell does not have any cell wall. Okay, so B, explain the role of carbohydrate in plasma membrane. Okay, so carbohydrate, okay, they are found by attaching to either protein that are found embedded in the membrane. So when the carbohydrate is attached to the protein, we call it as glycoprotein, or the carbohydrate can be found attached to phospholipid molecule in the membrane. So this one we call it as glycolipid. Okay, so these two molecules, glycoprotein and glycolipids, are important, or it involves in cell recognition or function as identification tag for the cell. Okay, so what is the significance of this feature or this role is that it is to allow cell to identify or differentiate themselves. Okay, so this is to make sure that the cells are grouped together. Cells of the same type are grouped together to form tissues, for example. Okay, or they also can be can form the antigen. Okay, antigen that are found on the plasma membrane of cell. Okay, such as antigen that are found on the surface of red blood cells that determine your blood type. Okay, for example, blood type A, blood type B, blood type AB. Okay, so your blood type is determined by the presence of different antigen on the surface of the red blood cell. Okay, so C, glucose, okay, is water soluble, while vitamin K is lipid soluble. So explain how each molecule crosses the plasma membrane by passive transport. Okay, so take note, glucose is water soluble. So that's mean, okay, the question basically asks you how each of these glucose and vitamin K crosses the plasma membrane. How does the two molecule enters into the cell, for example. Okay, so glucose is basically, it is a polar molecule. Okay, polar molecule. So because glucose is a polar molecule, so therefore glucose cannot pass through the phospholipid bilayer. Okay, they cannot pass through the phospholipid bilayer. So in order for glucose to enter into the cell, the glucose requires a transport protein, okay, to allow the glucose to enter into the cell. So in order for the glucose to enter into the cell, it requires transport protein. So this requires facilitated diffusion, or involve facilitated diffusion. Okay, so please add that one more point, where to cause glucose to enter into the cell, it requires or uses carrier protein or glucose co-transporter. Okay, and then the movement, because it is a passive transport, so the movement will be from high glucose concentration to low glucose concentration, or down its concentration gradient, or no energy is used or no ATP will be used during the process. Okay, so as for vitamin K, so vitamin K is a non-polar molecule. Okay, non-polar molecule. So because vitamin K is a non-polar molecule, they can easily dissolve. They can dissolve in the through the phospholipid bilayer or through the hydrophobic region of the membrane. Okay, so this causes vitamin K to easily pass through the phospholipid bilayer and through the process of diffusion. Okay, so basically vitamin K can easily diffuse through the phospholipid bilayer down its concentration gradient. Okay, from high to low concentration gradient, or no energy or no ATP will be used in the process. Okay, so altogether the total marks is 7 marks. So maybe 3 marks for glucose and 4 marks for vitamin K. Okay, any of these points. Okay, D, beetroot contains red pigments called betalains. Okay, betalines. So in an experiment, a student cut beetroot into cylinders and immersed them in 20% ethanol. After 10 minutes, the clear ethanol solution turned to red. Okay, so this example of question is basically what we have looked at during the lab, where you immerse turmeric, okay, into different concentration of ethanol. You have low concentration of ethanol and concentrated, okay, ethanol, and also the other one is distilled water. So you will observe at the end of the experiment, the intensity of the yellow color of the solution, okay, will be different. Okay, So same goes to this one, this question. This time you are using beetroot. Okay, so beetroot is red in color basically. So if you immerse the beetroot in 20% of ethanol, so at the end of the experiment after 10 minutes, the solution will turn red. So explain the changes in ethanol solution based on your knowledge on the structure of cell membrane. So for this type of question, it does not, it has nothing to do with hypertonic or hypotonic or isotonic solution. Okay, because for you to explain on that, it basically involves the movement of water molecule. But this one, we are looking at the movement of the pigment, okay, that causes the solution to turn red. So initially the pigment is in the cell of the beetroot. Okay, so you have to explain how does the pigment from the cell of the beetroot end up into this ethanol solution and causes the ethanol solution to be red. So here you need to explain on the movement of the solute, which is the pigment, the betalains. Okay, so by immersing the beetroot into 20% of ethanol, okay, so the ethanol will disrupt, okay, the structure of the cell membrane, okay, cell membrane or the plasma membrane. So this is because the phospholipid, which is the component of the cell membrane, will this is not dissolve in water but dissolve in ethanol. So please take note of this. Okay, the membrane. Okay, so I repeat, ethanol dissolve in, sorry, phospholipid dissolve in ethanol. Okay, not in water. Phospholipid dissolve in ethanol. So so therefore this will change the ethanol will also, okay, change the structure of the membrane protein. Okay, so protein that are embedded in the membrane, the structure of the protein will also change its shape. Okay, so the presence of the ethanol will also disrupt the membrane of the vacuole. Okay, so usually the function of the vacuole inside cell is to store the pigment betalains. Okay, so the betalain is inside the vacuole, so the ethanol will also disrupt, okay, the membrane of the vacuole and releasing the betalain from the vacuole into the solution and causing the solution to turn red. Okay, so initially the vacuole will have high concentration of betalain compared to the solution outside. Okay, so once the membrane is disrupted, the betalain will diffuse out, okay, diffuse out from high concentration of betalain to low concentration of betalain, causing the ethanol solution to turn red. Okay, so any of these 3 points, okay, worth 3 marks. Okay, 3 marks for each point. 1 mark for each point will total up to 3. Okay, so any 3 points. Okay, so number 2, when the concentration of ethanol is increased. Okay, so from 20% you increase, okay, the concentration to 90% by the student. So the red coloration become more intense. Okay, the red color will become much brighter. Okay, so what conclusion can you draw from this observation? So when you increase the concentration of ethanol, obviously it causes more damage to the plasma membrane of cell, or it causes the membrane to dissolve more. Okay, more of the membrane dissolves, and the membrane of the ethanol also dissolves and causes more pigment to be released, okay, from the cell and into the ethanol. Okay. Okay, so the total mark for question number 2 is 14 marks. Okay, question number 3. Figure 4 shows a model of structure of the plasma membrane. Okay, figure 4 shows a model structure of plasma membrane here. So explain why the model used to describe the structure of membrane is called as fluid mosaic model. So I have explained this earlier. So as you can see, one of the so these are the component of the plasma membrane or membrane, any membrane. It can also apply to membrane of organelles. Okay, so it consists of phospholipid bilayer, you have protein embedded in the membrane that have the different function, the 6 different function of the protein. So we call this as integral protein or transmembrane protein. And then you have carbohydrate attached to either of the phospholipid molecule or the protein molecule. So we call it as phospholipid or glycolipid and also glycoprotein. Okay, so why does the structure of plasma membrane called as fluid mosaic model? So mosaic due to the presence of proteins, okay, that are embedded in the phospholipid bilayer. So when for example, when you are looking from the top, it seems like it has a mosaic structure, okay, a mosaic pattern. And then for fluid, okay, so why does the name include the word fluid? Because the phospholipid molecule along together with protein, they can move, okay, they can move laterally, especially the movement is lateral, okay, lateral movement of phospholipid molecule and also the lateral movement of the protein, okay, molecule. So that's why it is called as fluid mosaic model. Okay, so B, explain why glucose cannot pass through the membrane by simple diffusion. Okay, so the major component, okay, of the plasma membrane is made up of phospholipid molecule, lipid. Okay, glucose is polar molecule. So glucose cannot just easily diffuse through the membrane. It has to enter into the cell, okay, through the membrane using transport protein. Okay, transport protein. So glucose basically is a large molecule. Okay, so glucose is also polar or hydrophilic, okay, or not lipid soluble. So because the glucose molecule is hydrophilic or polar, it cannot easily pass through or dissolve through the phospholipid bilayer, and therefore it needs glucose transporter. Okay, glucose transporter. So any of this point, okay, worth each one worth 1 mark, total up to 2 marks. Okay. Okay, question C. Okay, question C. Figure 5 shows the rate of glucose uptake into the cell across the plasma membrane when animal cells are exposed to different concentration of glucose. Okay, so as you can see from this graph, when you keep on increasing the glucose concentration, okay, so this will also increase the uptake of glucose by cell. Okay, so initially the glucose concentration increase, so does the rate of glucose uptake, but until to a certain point where it reaches a constant rate here. Okay, so based on figure 5, explain how the results of the investigation support the idea that glucose enters cell by facilitated diffusion. Okay, so as you can see, okay, from the graph, as the glucose concentration increases, you can see also the rate of glucose uptake increase. Okay, so glucose therefore will move down concentration gradient, can. So outside of the cell there is high concentration of glucose, so this causes the glucose to enter into the cell from high to low. Okay, from high concentration of glucose outside to low concentration of glucose inside the cell. Okay, so this increases the concentration of glucose inside cell. Okay, so therefore glucose move down concentration gradient from high to low, from outside cell into cell. Okay, so because okay, so this is due, or because as you can see, the rate keep on increasing because the presence of the transport protein or carrier protein is still available. Okay, so you have ample, or you have so many of the carrier protein that are still available to allow glucose to be transported from outside into cell. Okay, so the carrier protein is still available to allow the glucose to be taken up by the cell. Okay, but until at certain point, the rate of glucose uptake will become constant. Okay, will become constant. Okay, so constant. Why? Because the carrier protein, okay, the carrier protein that causes that involved in the uptake of glucose is now saturated with the protein. All is being used. Okay, all is being used at this point. So so this is when the graph reach its saturation point lah. Okay, so until equilibrium is reached. Okay, until equilibrium is reached, or until concentration of glucose, okay, inside and outside of the cell reaches the same concentration. Okay, reaches the same concentration. So 3 marks. Question D. Figure 6 shows a tube that acts as a cell in an experiment. The tube has small pores. Okay, so this is a tube, okay, that contains fluid. Okay, it contains fluid. So this tube will function as a cell. Okay, so the cell, as you know, it is surrounded by plasma membrane. So the wall of the tube will function as the plasma membrane. Okay, so the tube has small pores. Okay, small pores. So the pores will be like transport protein. Okay, so outside you have the fluid that function as extracellular fluid. Okay, inside you have the cytoplasm, which contain starch and also amylase. Okay, starch is complex carbohydrate, consists of glucose, and you mix the starch with amylase. So the function of amylase is basically it is an enzyme that breaks or hydrolyzes starch into smaller molecules. Okay, into smaller molecules, either as disaccharides or it can also become as glucose. Okay. Okay, so at the end of the experiment, fluid that acts as the extracellular fluid here, okay, the fluid outside here is taken to do some tests. Okay, some tests, which is it undergoes Benedict test, okay, and also iodine. Okay, so the extracellular fluid was tested with Benedict reagent and also iodine solution. So Benedict reagent is to detect for the presence of reducing sugar. Okay, so when you react, okay, when you mix starch with amylase, so the starch will be hydrolyzed to smaller molecule. So this smaller molecule can be maltose. Okay, so as you know, maltose is a type of reducing sugar. So when you mix maltose, okay, with reducing sugar, with Benedict reagent, so it causes the Benedict reagent to change color from blue to brick red, for example. Okay, so iodine is the solution to detect starch. Okay, starch. So the initial color of iodine is yellowish brown. So when you mix with starch, it will change to blue black. Okay, so for Benedict test, a brick red precipitate was formed. So this indicates it is a positive result that indicates in the extracellular fluid it has reducing sugar, either maltose or glucose. Okay, so for iodine test, the initial color of iodine, which is brownish yellow, does not change to blue black. So this means that in the extracellular fluid it does not contain any starch. Okay, as starch structure is large, so it cannot pass through the small pores, okay, that are present on the wall of the tube here. Okay, so explain these findings. Okay, starch will be hydrolyzed to maltose or glucose due to the presence of the enzyme amylase. Okay, enzyme amylase will hydrolyze the starch into smaller molecules such as maltose and glucose. So these two molecules, okay, they are smaller in size, so therefore they can pass through the small pores and enter into the extracellular fluid. So when you take the extracellular fluid, you mix with Benedict reagent, you heat the solution up together, so it causes the Benedict solution to change color. Okay, to change color. So this indicates that the the extracellular fluid contains the reducing sugar, either maltose or glucose. Okay, so maltose and glucose can pass through the small pores due to their small size. As for starch, due to the large size of the molecule, they cannot pass through the small pores, and therefore membrane is only permeable to glucose or maltose but not to starch. So therefore no starch is present in the extracellular fluid, okay, as it does not change the color of the iodine to blue black. Okay, so to conclude, okay, fluid contains only the reducing sugar but does not contain any starch. Okay, so any of these two points for two marks. Okay. E. Complete the table below by filling a tick in the appropriate boxes for the two transport mechanism. Okay, so requires ATP. Active transport, obviously it requires energy in the form of ATP. As for the osmosis, does not require any energy, so it is a type of passive transport. And then movement of water, osmosis, obviously it involves the movement of water. Movement of water does not require active transport. And then carrier protein are needed. Osmosis does not involve any carrier protein, but active transport does. Okay, channel protein are required for osmosis, which is aquaporin. And then molecule move down concentration gradient. Okay, so because osmosis is a type of passive transport, so it moves from low to high amount of water. Okay, so next, the next question number 4, give two components of plasma membrane. So plasma membrane, they have several components, so you just need to list down two, which is you can list down phospholipid molecule, or protein, or cholesterol, or glycolipid, and also glycoproteins. Okay. Question B. Name the transport protein for passage of water molecule across membrane. Okay, so passage, the transport protein requires by water, okay, to move across plasma membrane is aquaporin. Okay, so the answer will be aquaporin. Question C. Figure 7 shows a mechanism to transport sucrose and hydrogen ion across some membrane. So this one is the same as the previous question. Okay. So it's similar, not same but similar. So question number 1 here, name transport protein X. Okay, so this X is proton pump. Okay, proton pump. It pumps hydrogen ion from inside to the outside to concentrate hydrogen ion in the extracellular fluid side. Okay, so identify the active transport mechanism. So it is co-transport. So why do we call it as co-transport? Because this co-transporter brings in sucrose, okay, along together with hydrogen ion. So the significance of moving hydrogen ion from inside to the outside is to concentrate hydrogen ion outside of the cell, so it can produce a form of potential energy that the co-transporter can use to bring in sucrose, okay, from outside to inside, from low concentration of sucrose to high concentration of sucrose inside cell. Okay, so the question, explain the process in C2. So I've already explained. So these are the main point, okay, which involve proton pumps that pumps. Okay, so proton pumps that pumps hydrogen ion from inside to the outside of the cell. Okay, so remember, inside there is a low concentration of hydrogen ion, so that's why it requires energy from ATP. Okay, energy from ATP, from low concentration to high concentration. Okay, so this uses energy in the form of ATP, which then creates a concentration gradient or proton motive force. Okay, proton motive force is when you have high hydrogen ion outside. Okay, that needs to move back into the cell from high electrochemical gradient to low. Okay, to low electrochemical gradient. Okay. So hydrogen ion and sucrose, then together they will bind to the co-transporter. Okay, and causes the co-transporter to change shape and release these two substance, hydrogen ion and also sucrose, into the cell. So this one, okay, this means that the hydrogen ion moves down concentration gradient. Okay, moves down from high to low. Okay, and as for sucrose, move against concentration gradient from low to high. Okay, so this one occurs in plant. Okay, this mechanism occurs in plant so that the plant can concentrate sucrose, okay, or sugar inside of the plant cell, okay, inside the vascular tissue, for example, to be transported to other parts of the plant. Okay, from leaf to other parts of the plant. Okay, so next question, D. Explain what happened to red blood cell after being immersed in distilled water. Okay, so distilled water, I shorten it as DH2O. Okay, distilled water. So distilled water is actually hypotonic to the red blood cell. Okay, so because distilled water is hypotonic to the red blood cells, water will enter into the cell by osmosis. Okay, and causes, as water enters, it causes the red blood cell to swell and then bursts. Okay, for 2 marks. And then question E, explain the phosphorylation of sodium potassium pump. Okay, so sodium potassium pump, you need to understand it uses it uses active transport. Okay, active transport to move sodium, 3 sodium out and 2 potassium in. Okay, so phosphorylation means phosphate, okay, from ATP molecule will be will attached, okay, to the pump and causes the pump to change shape. Okay, that is phosphorylation. Phosphate from ATP will be hydrolyzed, okay, by releasing one of its phosphate and attached to the protein and causes the protein to change shape. Okay, so that is the purpose of phosphorylation, as you can see from this slide. Okay, so sodium will bind to the sodium potassium pump. So sodium from inside cell will bind, 3 sodium will bind, and then the binding will cause the phosphate to be hydrolyzed, releasing one of its phosphate and attached to the sodium potassium pump. Okay, so phosphate binds, or sodium stimulate the process of phosphorylation, the attachment of phosphate to the sodium potassium pump. We call it as phosphorylation. So the attachment of the phosphorylation causes the protein to change shape. So once the protein changes shape, so the binding site is now not compatible with the sodium ion. So sodium will be released to the outside. Okay, sodium ion will be released to the outside of the cell. So so this is how sodium is being moved out, okay, from outside to from inside to outside of the cell using sodium potassium pump. Okay, so explain, last question, explain how defective receptor protein on plasma membrane causes hypercholesterolemia. Hyper means high concentration of cholesterol outside of the cell. Your body cell is supposed to take up cholesterol, okay, cholesterol. But if the receptor on your plasma membrane is not functioning, okay, it's not functioning, or there is less or there is no receptor protein that can bind to the cholesterol to allow cholesterol to enter into the cell. So this can cause the cholesterol to accumulate in your blood. Okay, so which is dangerous. Okay, can lead to atherosclerosis. Okay, so cholesterol, okay, cannot bind. Okay, cholesterol cannot bind to the receptor protein. When cholesterol cannot bind to the receptor protein, cholesterol cannot enter into the cell and therefore accumulates in the blood and causes atherosclerosis or formation of blood or cholesterol deposition. So that's the end of question number 4, which worth 14 marks. Okay, so that's the end of discussion for chapter 3. Okay, thank you.